Basically, translation is similar in prokaryotes and eukaryotes. There are several key features that distinguish prokaryotic gene expression from that seen in eukaryotes. Let us watch a video on how translation termination occur in eukaryote first. Termination. Termination happens when one of the three stop codons is positioned in the A site. No tRNA can fit the A site at that point, as there are no tRNA that match the sequence. Instead, these codons are recognized by a protein, a release factor. Binding of the release factor catalyzes the cleavage of the bonds between the polypeptide and the tRNA. The polypeptide is released from the ribosome. The ribosome is dissociated into subunits and is ready for a new round of translation. Next, let's watch the video of translation termination occur in prokaryotes. Ribosome recognize polypeptide chain termination with the help of proteins called release factors. All RF reads the start codon and initiate series of specific termination events. E. coli has 3 RFs, RF1, RF2, and RF3. RF1 recognize UAA and UAG, while RF2 recognize UAA and UGA. RF3 does not recognize any stop codon, but stimulates the subsequent termination events. Elongation and ribosome translocation results in the stop codon entering the vacant A site. RF1 and RF2 binds to the stop codon which triggers peptidyl. Transferase cleaves the now complete polypeptide from its tRNA located in the P site of the ribosome. The tRNA is then displaced from the P site of the ribosome, followed by the disengagement of the RF and the 70S ribosome from the mRNA, and the separation of ribosome into the individual 30S and 15S subunits. Now, let's compare and contrast the translation termination in prokaryotes and eukaryotes. First, there is only one factor in eukaryotic cells called ERF1, whereas in prokaryotic cells there are 2 factors called RF1 and RF2. Next, for prokaryotes, only the formyl group from the first amino acid methionine is removed from the polypeptide after protein synthesis. While for eukaryotes, usually the unformulated first methionine as such is removed from the polypeptide after protein synthesis.